So if we could, we'll get started. It is my pleasure and an honor to be up on the stage, especially with Dr. Jarvis. But I think, run the risk a little bit of getting kicked off the stage since I'm in, the International Symposium on nicotine technologies, and I'm not going to talk about nicotine at all, though there is a slide in there that does have it, and we'll see if you can spot it. I'm going to talk about Kentucky Bioprocessing, which is a OpCo for Reynolds American. I am the Executive Vice President for R&D and for Scientific and Regulatory Affairs at Reynolds. And it's one of our operating companies, and my talk is entitled Elevating the Meaning of Transforming Tobacco, and that's what I'd like to talk to you about. I'm going to take you through a quick agenda. We'll go through the history and capabilities of KBP, some biopharmaceutical opportunities, how they might exist. Then I want to talk about KBP's response during crisis, also real world and rapid development, and then really what the benefits of that speed can bring. So for KBP, transforming tobacco is about making non-traditional products. So really, they use tobacco to make biopharmaceuticals to prevent diseases. In this case, Nicotiana banthamiana, which is a wild-type species from Australia. So I'll talk a little bit about the history of KBP. They're a global leader in cGMP production of plant-expressed proteins. They were established in 2006 in Owensboro, Kentucky. They have FDA and USDA regulatory experience, and they use transient gene expression and tobacco plants to produce high-value products. Reynolds worked with KBP for a number of years, but we ended up acquiring them in 2014. So to just give you some idea what their capabilities are, they have basically 3 acres under roof in an indoor greenhouse, which is fully automated and controlled. They can also do seed production. On the biology side, they can do protein engineering, which they have to do their expression. And then, of course, transient gene expression technologies. They use a couple of different methods to do this. From a manufacturing and purification standpoint, they have both lab scale, they have pilot scale, and then, of course, they have large scale capabilities. The lab, they can process about 10 kilograms of biomass. Pilot scale can process about 200 kilograms an hour, and then, of course, on the large scale side, they can process about 3,000 kilograms of biomass an hour. They're GMP compliant in terms of the purification systems, which are chromatography purification systems. And they also have a solvent processing room. And then, of course, they can do liquid, spray dried, and lyophilization, if I get that right, formulation, which is just a fancy word for freeze drying. So they can do all those formulations from their end products in isolation. They, of course, have quality capabilities. They have in-house method development, product stability chambers. They can do protein quality. Of course, on the QA side, they have cost-efficient GMP implementation. And of course, they use a hybrid approach between FDA and USDA type guidelines. Of course, they've been visited by FDA on a number of occasions, but of course, most notably in 2015 to review the ZMAP process, which I'm going to talk about here in a few minutes. So let's talk a little bit about biopharmaceutical opportunities and why the tobacco plant might be important in developing biopharmaceuticals. So biopharmaceuticals, unlike traditional drugs, are large molecules, right? So your traditional drugs are typically small, as shown on on your left. Biopharmaceuticals are large molecules or proteins, and they require much more complex means to actually manufacture them. And in fact, the standard in the industry is to use biological processes to manufacture these useful compounds. And there's a number of different systems that are used to actually do that. You can use transgenic animals, yeast. The most common way to make biopharmaceuticals is with mammalian cells, actually CHO cells. Chinese hamster ovary cells are most commonly used. And then, of course, you can use plants, like tobacco, for instance. But bacteria and other plant cells are also used. So why would you want to use tobacco plants to make biopharmaceuticals? Well, one of the key reasons is it grows really fast. It's also got a very favorable leaf-to-stem ratio, and what that means is most of the protein is accumulated in the leaves of tobacco plants, and the leaves are really big, which is very helpful because you can maximize the amount of product that you get out of it. Also, tobacco is very well known and very well characterized, right? It's one of the most studied plants in the history of man studying plants, and there's a lot of knowledge about how to grow it and how you can produce it. Another key aspect is it's not a food crop, right? So this is a crop that is not going to be in competition with either animal or human food chain, which makes it useful to study. It's not susceptible to human viruses, but it has minimal defense mechanisms, especially Nicotiana benthamiana, and we'll talk about why that's important in a minute. So how does KBP transform the tobacco plant? Well, it uses transient expression, which simply means that each individual plant is altered genetically by putting forth an insult to the plant, and then the plant actually reproduces whatever you insult it with. So if you're going to try to make a protein that'll be useful for a pharmaceutical, you express that protein in the plant, and then the plant just actually works like a bioreactor or like a photocopier and makes that protein and accumulates it in its leaves. And then KBP is able to knock down those leaves and extract it and purify that particular protein that you wanted it to grow. And so KBP uses a couple of different techniques to do this, and they utilize it depending on whatever type of product they're trying to make. So there's some strengths and weaknesses, of course, to making biopharmaceuticals with tobacco. Some of the weaknesses are there has not been an approved drug at this point in time that's been generated with tobacco. But some of the key strengths are manufacturing speed. This is a very rapid process compared to any other means that's used to make biopharmaceuticals. The plant itself grows, gets to maturity in the greenhouse in about 20 days, and then you can insult the plant, and then about 6 to 7 days later, the plant is ready for harvesting and then purification of whatever you put into it. So I'm going to talk about KBP's response to a global crisis and how they reacted to this crisis. And of course, what I'm talking about is the Ebola crisis in 2014-15. And I don't have to remind probably any of you that Ebola is a pretty nasty virus. It's a terrible disease. It's transmitted via body fluids. Mortality rates are very high, sometimes higher than 90%. And of course, it causes viral hemorrhagic fever, which is a very, very nasty way to pass away. It affects all the organs massively, of course, with very bad hemorrhaging. It's also been identified as a potential bioweapon if anybody ever figures out how to aerosolize Ebola. So that's another checklist on the concern. The crisis and pandemic in 2014 and 15 was the worst that had ever been seen. There were over 28,000 confirmed or at least suspected cases, over 11,000 deaths, and then of course, as I'm sure many of you know, this is the first time that Ebola actually escaped the continent of Africa, and there were cases brought over from workers in Africa to both Europe and to the United States. So KBP manufactured a product called ZMapp. And so what is ZMapp? It is a treatment for the Ebola virus. It consists of 3 monoclonal antibodies that target the Ebola virus. And each of these monoclonal antibodies were independently expressed in the tobacco plants, isolated, and then pooled together to make the drug product. And it was manufactured at KBP to do this. As you can see on the screen, we have CNN's Dr. Sanjay Gupta there. He's explaining what ZMapp is and kind of gets it a little bit wrong. Plus, I don't think he wanted to say the word tobacco on TV, and I don't think ever did. But he talked about really the proteins were expressed in mice, which they were initially when the research was started, but that's not how the product was made. So ZMapp got a lot of attention for this reason. Dr. Kemp Brantley, who was a doctor helping out with the pandemic in West Africa, contracted Ebola. And there was a course of treatment, a course of ZMapp treatment that was available at that time while he was sick. And he was given the first round of treatment, and he felt improvement within an hour. He then walked out of that African clinic and flew to the United States a few days later, and of course there's a lot of media coverage of that. And then he was admitted to Emory Hospital, given the remaining courses of treatment of ZMapp, and then was released a few weeks later, cured of the disease, or at least cleared of the disease. So I'll go through kind of the timeline of how ZMapp was developed, but I wanted to say to you that because of Dr. Brantley's use of ZMapp, or he consented to use ZMapp, KBP ended up manufacturing this product for a couple of years, and it turned out to be over 60 consecutive batches were made over a 2-year time frame under GMP conditions. The consistency as measured and monitored by FDA and others was of high quality. In fact, it even rivaled the quality of other types of products made in mammalian cells. And I did promise you that I thought I had one nicotine slide on here. There it is. It's at the very bottom. Part of the QA procedures was to make sure there was no nicotine in the ZMapp, and there it is, so there was no nicotine, or at least below level of detection. So let me go through what the timeline was on ZMapp development to give you some indication of how quickly KBP can move. The early products that were developed that led up to ZMapp was a product called MB003, and it consisted of 3 monoclonal antibodies that KBP worked on and manufactured, and it was pretty successful. In fact, the most successful treatment product that had been seen up to that time. But then our partners, MAP Biopharmaceutical, who owns the antibodies and helped develop the antibodies, came in December of 2013 and said, we think the 2 antibodies should replace other 2 in MB003, and we think it's going to be more effective. So KBP manufactured that in early January of 2014, made a small batch, which then went to a non-human primate study in Canada, and which was very successful in February and March 2014. And the set of animals that were exposed to ZMapp fully recovered, all of the animals, 100%. Even ones that were infected and got ZMapp treatment 5 days after infection, which hadn't been seen before. So there was a very effective means of trying to eliminate the Ebola virus. Well, August came around when Dr. Brantley got ZMapp as a treatment, and we had to make the decision at that point in time at Reynolds and at KBP to say we need to turn KBP basically out of the research function and just start making ZMapp, and we did that, kind of expecting that the U.S. government would come calling, and indeed they did. We signed a contract with the U.S. government to manufacture ZMapp and develop it. By October, we were done with our first campaign. We finished our second campaign in December, and then, of course, the U.S. government conducted a clinical study in West Africa starting in February of 2015. So that's pretty fast for drug development. And so what are the benefits of speed? Now, you can go to a bunch of different sites. This one, of course, comes off the Bill and Melinda Gates Foundation site to talk about pandemics. But if you're to model out a hypothetical flu-like pandemic like the one seen in 1919, just after World War I, that effectively killed about 30 million people worldwide. With no vaccine, of course, you have no countermeasures. With normal vaccine development, which usually takes about 30 weeks, you can obviously avert a number of deaths. But you can see where we're going here in terms of speed. If you can cut 8 weeks out of the normal vaccine development period, you can significantly cut into the amount of damage that the pandemic does. And then, of course, if you can get down to 6 weeks, you can virtually eliminate almost all of the deaths. And of course, 6 weeks is whereabouts KBP is operating in. In the 6 to 8 week time frame is what we would predict we can do. The plant grows within 20 days, we infect it, wait another 7 days, then you process. You're in a very, very short time frame. So speed is very, very important. So I'll conclude by simply saying this. Today, tobacco is generally made to make pleasure products like cigarettes. And really, in terms of transforming tobacco, what we're looking at is having tobacco, and companies like KBP transform the plant into generating things like biopharmaceuticals or other useful products for human benefit. So that's the conclusion of my talk. I'm not sure if I've got time for questions, but I'm happy to take some or talk to y'all outside.
Thank you very much for a fascinating talk. We're taking us outside our comfort zone, I think. But I'm sure the FDA would have been very pleased to see the zero nicotine, because that looks like it could be really useful for reduced nicotine cigarettes. Any questions, please, for Jim?
Sorry, you showed tobacco being grown hydroponically and under grow lights. I mean, one of the broader aims when talking about kind of shifting away from using tobacco for products that people can smoke is to find other ways for tobacco farmers to have a livelihood. Would it be possible to grow these KBP type tobacco products outside? I mean, in nature, or is that really not feasible for complying with GMP and so on?
No, it is feasible, and in fact, KBP has done that. They work with farmers to do that. There are complications. You don't have the control that you have inside the greenhouse, and you said they're not exactly grown hydroponically. They do have a soil matrix, but it is a lot easier to control both the environment and the conditions, and that helps maximize the speed, because if you're left to the vagaries of nature, sometimes you can't depend on the plants growing the way that you want them to. But there are other applications that KBP and Reynolds have worked on that would take the farmer in mind for other products that I can't exactly talk about right now. But indeed, our interest in KBP is they are experts at purification and extraction, and we've been working with them a long time to do exactly that.
In terms of the choice of using tobacco or mammalian cell culture systems to manufacture monoclonals, is there a species or an end where tobacco is more or less applicable, or is it completely up to the manufacturer to decide which one they could use?
Yeah, I think it depends on the scale, right? So if you take a product like ZMAP, which was really a very difficult product to make, and in fact, the dosage of ZMAP was actually in the grams level, okay? So that is a pretty heavy dose of active ingredient to have to take in to fight this disease. And of course, even though the tobacco plant expresses the proteins actually quite well, having a dosage requirement that high may not be the best utilization of at least KBP's current facility. If you had much larger facilities, it would be better. Of course, if you go down in dosage and you talk about things like vaccines, for instance, which are in the microgram level, of course, the tobacco plant works very, very well for those types of systems because you can make quite a lot of it and have a lot of doses. Does that answer your question?
Yeah, it does. Yeah, thank you. In terms of yield, then what is the percentage yield on?
It depends on the product. So,
but for the ZMAP, for example,
I can't remember the exact. I can't remember exactly how much it expressed, but we were getting about. We were not filling up the greenhouse, but we were getting about 100 doses a month, courses of treatment. I think of that. So not great. If you turn that into, something at the microgram level, of course you've got millions of doses.
Thank you.
Well, thank you very much indeed for your talk.
Appreciate it.